Alrighty, hello everyone, and welcome to July's Life RCN seminar. We are very excited to have you all here, and we are very excited for our speaker today. Our speaker today is Dr. Paula Welander. She is an associate professor in the Department of Earth System Science at Stanford University. She received her undergraduate degree. From Occidental College in Los Angeles. She pursued her PhD in microbiology at the University of Illinois at Urbana-Champaign. And she completed her postdoctoral studies at MIT in the departments of biology and of earth science, earth, atmospheric, and planetary sciences. She joined Stanford faculty in 2013, where her current research program is focused on understanding the biosynthesis and physiological function of molecular fossils, which we'll be hearing about today. And take it away, Dr. Welander.
Great. Thank you, Jaime, for the invitation. And thank you, everyone, for joining me. I'm going to go ahead and share my screen. Okay. Let me get rid of the... Sorry, I'm just making sure I'm putting everyone in the right spot here. All right, great. So can everyone see that? Did it start presented? Okay, great. Jaime, we're good?
Yep, we're good.
Okay, great. Thank you, everyone, for joining. So today I'm excited to share with you the work that we've been doing in my group on membrane biosynthesis in Archaea. So my group, very broadly speaking, is very interested in lipids, and we are interested in them for a couple of different reasons. Well, one, lipids can be divided into very diverse groups of different types of lipids that are present out there, and they also have very diverse functions within the cell. So lipids are important, most famously known for their membrane and the work that they do in maintaining cells and protecting particularly microbial cells from their environments. But membrane fluidity and rigidity is really dependent on different types of membrane structures and lipid structures. Lipids can also be precursors to a lot of important molecules within organisms. For example, within humans, the hormones are precursors, lipids are precursors to our hormones. They're important in storage, both for microbial organisms and storing excess carbohydrates as fats, and also for us, we store lipids as well. Lipids are also really important in signaling, and particularly in eukaryotic organisms and in multicellular organisms in terms of being able to, in development and these types of signaling pathways. Lipid transport is very important in human disease. The inappropriate transport of lipids in different areas results in a lot of diseases that are difficult. And finally, they're also really important in pathogen interactions. So a lot of times, microbes, bacteria that infect human cells are able to utilize lipids as a source of carbon. They're able to utilize lipid pathways as a way to circumvent immune systems and be able to really mount a pathogenic attack. So lipids are not only varied in the structures and the types of molecules we can find both in complex organisms and in simple cellular organisms, they're also very complex in the types of functions and roles they play in these cells. In addition, and this is probably what I'm more well known for in thinking about lipids, and it is actually the area that got me interested in thinking about lipids, is these molecules are also very well preserved in the rock record. So you can have lipids such as cholesterol that through the process of diagenesis, which is the process of how a sediment becomes a rock, can be preserved in the rock record in these basic structures shown here. And this is important because if we look at the ability to preserve molecules over time, so this is showing you the history of the Earth from when the Earth arose 4.5 billion years ago to today. You can see the preservation potential for a lot of molecules. For example, DNA and proteins are only preserved in rocks for hundreds of thousands of years, where lipids can be preserved over a billion years. And in particular, for organisms like microbes that don't generally leave fossils laying around, these molecules can be indicator for these types of organisms. And so based on that, there has been a lot of work in the biomarker field based on lipids, looking at how, what organisms produce these different molecules and how they're preserved in the rock record. And so, for example, we know dinoflagellates produce this 4-methylstyrene that can be preserved in the rock record. And because of its occurrence in these organisms, it's used as a biomarker for those organisms. And so studies like that and correlations like that have led to generating these biomarker trees of life, where different biomarkers can be used very broadly as indicators of eukaryotes or bacteria, or very specifically of certain types of organisms and their metabolisms. And so it's in this area that my research group is really interested in contributing. We want to be able to aid in interpretation of these biomarkers, even if we're not the ones looking for them in the rock record. And so in my group, everyone kind of has their own lipid that they work on, and they're different organisms. And we're interested in them from the biomarker perspective from some projects. And for some projects, we're just interested in them from the basic physiological function of them as well. And hoping that all that information will really help us understand the diversity of lipids in different types of microbes, as well as the diversity of function, and as well as the potential to be used more robustly as biomarkers. And so today I'm going to share a story with you, a couple of stories actually, on our work on looking at Archaeal ether lipid biosynthesis. And this is a project that my graduate student, Andy Garcia, is currently working on, but was initially started by a former postdoc in my lab. Okay, so why are we interested in Archaea? So I love Archaea. Archaea are very interesting microbes. And primarily they're interesting because I think they're a little sneaky. So we were studying Archaea for many, many years. Particularly methanogens were discovered in the 50s for their ability to produce methane gas. And they were studied. And when you looked at them under the microscope, they looked just like bacteria. And so for many years, these types of organisms were considered bacteria. And then in the 1970s, Carl Woese, who was a professor at the University of Illinois, pioneered the use of 16S ribosomal DNA as a phylogenetic marker, a way to trace microbes phylogenetically, a way to kind of start to assign evolutionary relationships between different types of microbes. And through this work, what we saw was that actually these organisms that we thought were bacteria were actually a complete different branch of on the tree. They were no more closely related to bacteria than they were to more complex organisms like eukaryotes. And he named these organisms the Archaea. And so these organisms have been studied for many years now, and there's a lot of distinctions besides the phylogenetic distinction from bacteria and eukaryotes. And one of those things that distinguishes them, that interests us, is their lipid membrane structure. So Archaea are distinct in that their membranes have a similar overall structure to what we see in bacteria and eukaryotes, but the actual chemical composition is different. So for example, they have these hydrophobic tails that are linked to a hydrophilic head. But what we see here is that in bacteria and eukaryotes, the tail is fatty acids, ester-linked to a glycerol 3-phosphate backbone, whereas in the Archaea, these tails are isoprenoid chains, which are completely different biosynthetic pathway, completely different type of lipid. They're ether-linked, which is a much stronger connection, and they're linked to a glycerol 1-phosphate backbone rather than glycerol 3-phosphate. In addition, the Archaea do this very interesting thing with their membranes, and they produce these GDGTs, which is basically taking two of their diethers and fusing them to form this tetraether that we call GDGT. And so what essentially they end up doing is creating this bilayer into a monolayer. And so this is what the membranes look like. And this can be up to 90% of an Archaea cell's membrane composition can be fused in this way. And if that wasn't strange enough, they also add these rings into these GDGTs. And so they can add pentacyclic rings to produce GDG4, GDG8, and they can even add hexane rings here. So the Archaea are interesting. They're a completely different group of organisms from bacteria and eukaryotes, and they make these weird lipids. And so why do we care about these? So in my group, we care about these for two reasons. The first one is there's a potential question that arises from this structure in thinking about the phylogenetic and evolutionary history of eukaryotes and how eukaryotes arose. So when Carl Woese discovered the Archaea, he suggested there was 3 domains of life: the bacteria, Archaea, and eukaryotes. And for many years, as more Archaea were discovered or sequenced or cultured, this structure of the domain of tree of life was pretty robust. But in 2015, there was the discovery of this Loki Archaea, and then more recently, a larger group of these Archaea known as the Asgard-eukaryote that through phylogenetic analysis started to kind of compress this tree into 2 domains of life, where you have bacteria and Archaea being the main domains of life on the tree, and the eukaryotes branching within the Archaea and even emerging from the Asgard-Archaea. And as more sequences of these Asgard-Archaea have been discovered, we are seeing that, that this is holding true, that you have these two domains of life, the bacteria and Archaea, and that the eukaryotes are nestled within the Asgard-Archaea. And I mean, this paper came out from Brett Baker's group and Tij Ettema's group just in June. So you can see as we're getting more and more sequences, this idea of a two-domain tree of life is robust. So why is this significant from a lipid perspective? Well, as I told you, the Archaea have different membranes from the eukaryotes and the bacteria. And so if you have this idea that the eukaryotes emerge from the Archaea, they have completely different membrane structures. And so there's this hypothesis that there had to be a membrane transition. And so if you can imagine, you have the most ancestral organism, both to bacteria Archaea, and we are not sure what the membrane structure of that ancestor was, but we know that there was a split between the bacteria and the Archaea. And we can imagine that this is perhaps where the membrane structure split. And somewhere along this line, the Asgard-Archaea evolved. We have no idea what their lipid structure is. And then the eukaryotes emerged from within the Asgard with the same structure of lipid as the bacteria. And so this becomes a problem when we don't understand how this occurred, if we don't know or understand the lipids of the Asgard-Archaea and understand the mechanisms behind the lipids of Archaea more broadly. Now, there finally was one Archaea that was, actually there are two now in culture, Loki Archaea or Asgard-Archaea that are in culture. One of them in this study, they did the lipid structure and they found that they had the isoprenoid-based ether-linked membrane lipids that we see typically in Archaea. So in this one instance, we see that there is this, that this Asgard-Archaea has this traditional archaeal membrane. And so again, we have this transition here that we can't really explain or understand. But this is one organism, and we really don't know what the structure is of all these other uncultured Asgard-Archaea that might provide insights into how this transition occurred. So it's really important for us to not only understand the lipid structure within the Asgard-Archaea, but also just broadly in Archaea. As you'll see from this talk, we really don't know a lot about the biochemistry even behind the membranes of these unique organisms. So that was one reason that we're really interested in. The other reason we're interested in these lipids is from the biomarker perspective. So these GDGTs are also well preserved in the rock record. They can be preserved about on the scale of hundreds of millions of years. And in the early 2000s, it was proposed that these cyclizations can be used as a response to temperature, and that you can link that to sea surface temperature in the past. And the way this is done is it was demonstrated that when they measured the ratio of different cyclized GDGT in marine sediments, there was this correlation to sea surface temperature. That is, the ratio increased, the temperature increased in the environment that they were looking at. And this is a very powerful tool that can be used to, if you then look in sediments and find fossilized GDGTs that are cyclized, you can apply this proxy to this and make predictions about what the temperatures, sea surface temperatures were, several hundred million years ago, which will then feed into climate models, and you can make inferences about past climate events that can then be modeled into future climate events. It's a very powerful tool in paleoclimate studies. And over the years, these other proxies have been developed. This TEX-86 proxy was the first one that came out with GDGTs, but there have been modifications made based on reanalysis and recalibration. More data is collected from the environment. We get culture data and people start reassessing these. And so these GDGT-based proxies seem pretty robust. But like any proxy and like any biomarker, you're also making certain assumptions. And this is where we felt that we could contribute. And so one of the assumptions that is made in the TEX-86 proxy is that there is one major source, microbial source, of cyclized GDGTs in the open ocean. And these are these Archaeota, which were previously called Marine Group 1. So you'll see me interchangeably using these terms. So these are organisms that produce these GDGTs in marine environments. They're ammonia oxidizers. They're very slow growers. They can take a really long time to grow. Several species have been identified, and some of them have been studied significantly. But again, they're very slower. There's no genetic system, so they're difficult to work with. And they're not just found in marine environments. Turns out that they're in all across different types of environment, both terrestrial, freshwater. But they're the only archaea known to produce specifically this molecule, Archaeal, which is part of the TEX-86 proxy. So this was assumed to be the major source of cyclized GDGT and the major source that was being based for the TEX 86 proxy. But in 2014, there was this paper that came out that using 16S analysis in the Pacific Ocean across depths here, going from 0 to 600 meters, demonstrated that there was another source of Archaea in the ocean that was more dominant at upper depths. And these were these marine group 2, which are Eukaryote. And at the lower depths, they showed that the marine group 1 were more dominant. And so why would this be an issue for the proxy? Well, multiple sources could distort the signal. So if you can imagine what the signal for the TEX 86 signal is showing that the Archaeota are modifying their membrane lipids or changing the cyclization in response to the temperature. But if you have multiple sources, that change that you're seeing in GDGTs could actually just be a change in the community rather than actual change within the cell of the temperature. And that can kind of skew the signal. The other problem we have is that the marine group 2 Eukaryote have not been cultured. So we don't actually know what the lipid profile of these organisms are. And this distribution here is based on 16S ribosomal RNA gene data. So it's telling you that there's more of these marine group 1 here versus marine group 2 here versus marine group 1 here. But that doesn't indicate what the lipids are, right? Because the lipids would have to be based on actual gene data from the lipids, not from the 16S ribosomal RNA gene. And so what we thought was to properly resolve the cyclized GDGT source issue, that we needed to look for GDGT cyclization proteins in these environmental data sets rather than looking at 16S. And the problem we have is that at the time, we didn't know what those proteins were. And so this is where my postdoc at the time, Ray, picked up the project. He's now a professor at Southern University of Science and Technology in Beijing, China. And we did this project with 2 of our collaborators, Shiloh Lee and Roger Summons. And so the question we had was very basic. Can we identify the proteins that add the rings to this GDGT molecule and then look for that protein within metagenomic data sets in open oceans to see what the predominant source are at the different depths? And when we looked at the literature, there really was no hypothesis about how these rings could be added. This is a very inert molecule, and it would have to be activated. And so in looking at the literature, we kind of took a leap of faith and said, well, maybe this involves a radical mechanism. And if it involves a radical mechanism to activate these carbons, that most likely involves a specific type of protein known as a radical SAM protein. So what is a radical SAM protein? So these are these proteins. It's a huge family of proteins that do all kinds of different chemistry. But what I'm showing you here is this amino acid motif, this cysteine, these 3 cysteines interspaced here that are responsible for helping activate a lot of inert carbons. And so what these cysteines do is they will take this adenylyl, deoxyadenylyl L-methionine, or SAM. This is a very common cofactor in a lot of biological reactions. And these cysteines will split this into this 5' deoxyadenylyl radical. And it's this actual radical that is able to attack these inert carbons and really be able to catalyze a lot of really hard chemistry to catalyze a lot of hard chemistry. So we decided to look for this GDGT ring gene in Cephalobis acetocaldareus. So this is not a Archaeota. This is not a marine organism. This is a hyperthermophile from a hot spring. And it's acidophilic. It goes at about pH 4. And we chose this organism because it produces GDG with rings, up to 8 rings, and it had an experimental system established. So we assumed that any genes that we found involved in GDT synthesis in this organism would be true for other archaea that make GDG rings. So this is an assumption we had to make. And the way we attacked this was we looked for all the radical SAM proteins in the Cephalobis genome. We found 18, which is a very, very manageable number. It's a very low number, actually, of radical SAM proteins. And what Ray did was he did a comparative genomics where he looked in which radical SAMs were present in ring producers, which ones were not in strains that didn't produce rings, and he got these 3 candidates. And so he went ahead and did deletion mutants. So here I'm showing you a chromatograph of the wild type, showing that it makes, in our hands, it made up to 6 rings. And when he knocked out one of these proteins, this 1585, he lost almost all ring production right away, which was really exciting. But we still saw the production of one ring. And so he started to kind of try different combinations of these three candidate proteins. And when he knocked out this 1585 and this 240, that's when he completely lost ring production. And so we did some complementation studies to confirm this and ended up naming these proteins GDG ring synthesis, GRSA for the 1585 and GRSB for the 0 to 40. And so we did a lot of experiments on how GRSA and B are generating these rings. I don't have time to go into all of those experiments, so I'll just give you kind of the gist of the story. We found that GRSA was adding rings at the C7 position, and GRSB was specific for adding at the C3 position. We also found that this reaction was sequential, that GRSA preferred to add rings to GDGT0, and GRSB preferred to add rings to GDGT4, so something that already had 4 rings. But having found these proteins, we really wanted to answer the question about what the source was in the open ocean. So we went back to this paper and found their metagenomic data set, and there was a total of 38 metagenomes available in the GGI database. And so we wanted to identify our GRS homologs in these metagenomes. And so we went ahead and did that, and we found over 1,000 homologs. Some at this 100-meter depth, but the majority of them were very deep within their samples. And we took these sequences from these different depths and we put them into a larger GRS tree with cultured Archaea, so that genomes, so that we could see where these were landing, what were the source of these. Were these Eukaryote, were these Archaeote? And so the tree we generated is this huge tree here, which I will zoom in a second, but I wanted to orient you a bit. So here in blue are the GRSAs from Cephalobis that are homologs to the ones from Cephalobis, and GRSB, again, homologs to Cephalobis. And all the sequences we found are in the metagenome sequences are shown here in red. So I'm going to zoom in, and what you can see here is that all of these ones from these metagenomes that we found are clustering with Archaeote, with marine group 1. None of them cluster with any other Archaea from across that tree that we have. In addition, when we were about to publish this paper came out that reported 270 MG2 metagenomes. And these were from various marine environments. So these were, and we didn't find any GRS homologs within these genomes. So either these Eukaryote do not produce cyclized GDGTs, or if they do, because again, they're not cultured, so we're not really sure, they're using a different mechanism. But given the data that we have now and what I've presented to you here, it seems that marine group 1 are the major source of cyclized GDGTs in the open ocean, which helps us constrain this TEX86 marker a little bit. And now we have a tool where we can go out and look for these in these other metagenomic, metatranscriptomic, and lipid data sets to try to ascertain if this is just true in this data set or is it true across other data sets in marine environments that could potentially help us constrain this proxy a little bit more. Okay, so the work that we did with GRSA and B was really exciting and got us interested in thinking broader about some of the unanswered questions that were present in GDG biosynthesis. And so, oops, wrong way, sorry. So one of the questions we still don't know is how or what proteins are involved in generating this GDGT0, so how this condensation reaction happens between 2 diethers to generate a tetraether. Some of the other questions that were unknown is we knew that there was this geranyl-geranyl reductase that could remove the double bonds, fully saturate to generate these diethers shown here, but it was unclear if this happened before or after condensation. And then it was unclear whether GRSA and B needed the double bonds, the proteins that we worked on, to actually add the rings. And so when Ray was finishing up in my lab, he was interested in starting to understand how this reaction might occur here. So we hypothesized that a radical mechanism would also be necessary for this. And we focused on 15, we had identified some other, there were 18 radical SAMs in Cephalobis, and we had identified some for other functions. So we were down to 15 radical SAMs potentially. Ray found this Saki 0703 radical SAM, which was the most promising candidate based on comparative genomics, on what has GDGT, fused GDGs and what has only diethers. But also he found a homolog in this Methanercyanus species here. This 1486 is a homolog to this radical SAM. It's right next to the geranyl-geranyl reductase. And so Ray was just really convinced that this was the one that was occurring. The problem we had when he was in my lab was he would try to make this deletion and Cephalobus just died. He could not get this mutant. And so we thought this was essential. But at the same time, something that bugged me was that this protein in this methanogen, so it's a homolog to this protein that we think does this condensation, had been shown through biochemical studies to add these methyl groups to this methanotarian, which is a cofactor in methanogens. And so to me, it was like, this already has a function. And so it's essential, it has a function. And I was convinced that Ray was wrong. So Ray took this project to his lab and started his new lab with this project and immediately proved me wrong. So I lost this bet with Ray. But the problem Ray had was that he couldn't express this protein from Cephalobus. He couldn't delete it. And so what he did when he got to his lab was he developed a system in methanogen to express proteins. And it was a methanogen that only made the diethers, didn't make the tetraethers. And then he couldn't get this Cephalobus protein to work in the system, but he found the homolog in another methanogen that did work. And when he put it in this organism, he could then get it to produce this weird microcyclic Archaea, GTTG, and then GDGT. And so he ended up proposing this pathway where you have this tetraether synthase, is what he's calling it, this Saki 073 homolog that can generate an intermediate and then generate GDT0, and an off product is this microcyclic Archaeal. Subsequently, Squire Booker at Penn State, along with his graduate student, Cody Lloyd, found the structure independently of this, published the structure of the original methanogen one that had that other function that is wrong. And what they found in this beautiful paper, it's a really beautiful study, was that it could actually bind, that when they crystallized it, they found it bound to fully saturated Archaeal or architylglycerol. Indicating that most likely the mechanism of TES, or what they were calling GDGT-MAS, is on the fully saturated Archaeal. So that seemed to resolve that question, or at least provide a little bit more evidence. Some people still don't agree with that mechanism. So what we think is happening here, what we've been able to resolve so far, is that we have this protein GGR that is able to fully saturate. TS then forms GDGTs, and then GRSA and B add the rings. So one thing that struck me was that we could show that GRSA and B were non-essential, but so far we had been able to generate mutants. So Ray tried to generate this mutant as well in Cephalobus and this mutant, and haven't been able to generate those mutants. And so we had this question about some of these proteins being essential, some of them not being essential. And so this is where my student, Andy, who came in and started to think about different questions that they wanted to address in GDGT biosynthesis. And he came across this paper that had shown that in Thermococcus codocarensis, which is a thermophile, it's an anaerobic organism that grows at neutrophilic. So unlike Cephalobus, it's not an acidophile. But this paper in 2019 had done this random mutagenesis and generated a mutant randomly in this 2145, which turned out to be a homolog to the Cephalobus TES. And when they looked at the growth phenotype, so this paper, they don't know what this protein is doing, but they showed that at 85 degrees, this mutant, this TES mutant grows fine, which is the normal temperature for Thermococcus to grow. When they increased the temperature, they saw a slight growth defect shown here. And so this is why they picked up this random mutant. But then the paper, they just left it as a hypothetical protein, and Andy made the connection that this was a TES homolog. So Andy wanted to pursue this and see if this actually was an essential protein. And Andy also wanted to bring in this organism, Thermococcus, as a model organism for us to work on in our lab, because it's actually very well established in terms of genetics. And Tom Santangelo's group at Colorado State University is really the world expert in doing genetics in Thermococcus. And so we reached out to them and together started a collaboration with Ali and Kristen and Tom, where Andy went and spent a week there, learned how to work with this organism. And the question we're trying to address is, are these GDGT proteins essential in this organism? And so the first thing that Andy did was he got some mass, some biomass from the Colorado group, and he measured the lipids. And of course, we could see that it can make the diethers. It made Archaeal, and it made a little bit, tiny bit of this macrocyclic Archaeal, which seems to be a side product. Andy, of course, found the TS homolog. He also saw a GRS homolog. So they thought there should be cyclized GDGTs. When they measured the GDGTs, the majority, like 99.9% of the lipids were at 85 degrees and 95 degrees were GDGT0. So the TS is definitely active. But when they looked, within like less than 1%, they could see rings. And when they grew them up at 95, they could see a little bit more, but not much more. So this organism does make GDGTs with rings. It's been reported to make minor amounts in the literature, and that's what Andy was seeing here when they did the lipid analysis. So we collaborated with the Colorado group, and they made the mutants, and we're able to show that you could knock out TES and get rid of GDGT0. So here's the wild type showing the diethers and the tetraethers, and you can see it makes Archaeal and GDGT0. When they knock out the TS homolog, we still make Archaeal. We don't see any GDGT0, and that is returned. We do see GDGT production restored when we provide TES on a plasmid. They also knocked out the GRS protein, and he did the lipid analysis. And what we can see here, like I said, unfortunately, this organism makes very, very small amount of lipids, so the chromatograms don't look great. But we can see if we extract the ions for the different rings in the wild type, you can see it makes tiny amounts of 1 through 4. That's completely lost in the GRS mutant, and then it's restored when we put a copy, nectopic copy in on a plasmid in the mutant. Okay, so these proteins are doing what we think they're doing, the same thing that they do in Sophilobus, but we're also showing that both TES and GRS can be deleted in this organism. So what are some of the growth phenotypes that we saw? So we saw some interesting growth phenotypes. First, if we got rid of GDGTs completely, you could see that at 85 degrees, there was a slight growth defect. So you can see in red, here is the TS mutant, and then it hits stationary phase, and we see a significant decrease in, we see death. That's dramatic, and that's that phenotype is lost when we complement the strain. You can see here in blue that it no longer crashes in stationary phase. At 95 degrees, when there are no GDGTs, we would see, which is, again, this is the temperature, high temperature. This is normal temperature for Thermococcus. There's just a longer lag phase here. It takes a little bit longer for it to grow up, the mutant. And then again, at 95, we see this crash, though not as dramatic in stationary phase. So there seems to be some role for these GDGTs in survival at higher temperatures during stationary phase. For the GRS mutant, so this had a very interesting phenotype. So this organism is making GDGT0, but it's not making any cyclized GDGTs. And at 85 degrees, we saw no growth phenotype. It looks perfectly fine. And we also didn't see a growth phenotype in red. So this is the mutant in red shown here at 95 degrees, but we saw this weird phenotype when we complemented. And that is that it had this lag growth. It eventually reached the same OD, but it did grow a lot slower. And this was very pronounced. And so we started to think what could be happening here when we see this in overexpression. So Andy decided to look at the lipids more in detail. And what he saw was, if you look in the wild type at both 85 and 95 degrees, as I showed you earlier, the percentage of cyclized GDGTs is like very, very low. But when you overexpress this protein, or when you complement, we didn't measure we didn't do Western blot. We're not sure how high the overexpression is, but when you complement, we're assuming there's a lot more copies. You make almost 30% of the lipids have rings. And that's a huge difference for this organism, right? It goes from making a very, very small amount of rings to now having all these rings. And maybe that's what's causing this delayed growth here. So in addition, Andy also saw when he looked at the diethers in particular, so here's the wild type at 2 different temperatures, you don't really see anything but Archaeal. But here you could see these, what we are calling Archaeal 1 and 2. It looks like GRS was able to add rings to the diethers. And we don't see these in the GRS mutant, so we're pretty confident they're rings. And we don't see these in the wild type. And so we think that maybe we've altered the lipid profile enough to cause that defect to the point where the cell is not growing as well. And so what I think we're showing here in Thermococcus and being able to make the mutants is that there is some kind of interesting complex interconnection between these proteins. So if you can imagine, TES is able to make this microcyclic archaea on the side. And if we delete GRS and you're no longer able to add rings, maybe you start to add more macrocyclic archaea to the lipid. We offset the balance. I think the same thing can happen when we knock out TES and GRS no longer has the GDGT substrate, and now it starts to cyclize other lipids. And so we think there's some kind of regulatory control that would be really interesting to understand here between these different proteins and how they interact. The other thing that was really exciting about this is that for Andy, this really became a model system that we could use for heterologous expression. And so one of the things that Andy first started to think about when they first joined my lab was looking at the GRS A and B proteins and their diversity in the environment. So this was the project that Andy first started, because Andy started during the pandemic. And I first wanted Andy to just do a bioinformatics project. So I was like, look at these proteins. And Andy went after and looked in metagenomes and genomes and generated this phylogenetic tree of GRS homologs across genomes and metagenomes. And when he collapsed this so you could see the different kind of clades that are here, what he found was that there are like 10 clades of potential GRS homologs. And then GRS-A from Sophilobis is in this one clade, and GRS-B is over here. But we have all these other clades with potential GRS-B proteins, ring synthesis that we actually don't know if they're ring synthesis. It was like we didn't know where the cutoff was here. And a lot of these are from uncultured organisms, from metagenomes or metagenome acquired genomes. And so we don't actually have organisms we can test to see what their lipid profile is. In addition, Andy got in contact with Valerie, who's a postdoc in Brett Baker's lab, and they have a large data set of Asgard-eukaryote, and we were able to see that there are Asgard-eukaryote distributed throughout the tree that potentially could have been making these GRSs. But again, we don't have these in cultures, so we need a way to verify that these proteins are actually GRSs. And so this is where we thought we could potentially take this new Thermococcus system we have working in a lab and see if we could get expression to work. And so to begin with, Andy started by looking at this clade 3. And they were interested in this clade 3 because it's dominated by Tom Archaeota, and Tom Archaeota are the only Archaea known to make this crane Archaeal. So we thought maybe some of the GRSs within that clade could potentially be not only adding GDGTs, but could be adding this, could be adding the pentacyclic, but maybe the hexic ring here as well. The other thing is that we had noticed is that in the Tom Archaeota genomes, the GRS copy number differs by habitat. And so in the marine Tom Archaeota and organisms like Nitrosophilus maritimus, there's like 2 copies of GRS, which is you would expect if they're making this kind of a lipid, right? 2 GRSs, one of them is adding these different rings. But in the hot spring Tom Archaeota that have the same lipid profile as the marine organisms, we're seeing they can have up to 6 copies of GRSs. And we're not sure if any of those which ones are functional, are they all GRSs? Some are just redundant, and we don't know what's going on there, or why those that are come from hot, more thermophilic environments are actually had that many copies. So this was, we thought, a good way to kind of start to address this, particularly because these organisms are hard to culture. So initially, Andy began by trying to express some of these in Sophilobis, and they expressed them in the GRS mutant, and we had no expression. We couldn't get this to work. And this is one of the reasons why we were kind of motivated to look for new systems to express these in. And we thought, I mean, even though this was a thermophile and we thought the GRS would function in this, and Sophilobis is an acidophile, these other organisms are not, so we thought that might be a problem with expressing the protein. And then Andy went and tried this expression of this one GRS from this Tom Archaeota, and was able to get expression, was able to produce all of GDG 0 through 4. And so what you can see here in the Thermococcus GRS mutant produces a lot of GDGT0, and we see no cyclized GDGTs as expected. And when Andy puts the GRS from this Tom Archaeota into this mutant, we now see production of cyclized GDGTs. So the heterologous system seems to be working, which is great. Andy went on and measured this and actually showed that even though the percentage overall is small, it's about 2 to 3%, that's a lot more than Thermococcus produces itself, right? Like under normal conditions, the wild type, I said, again, produces less than 1%. And here we're seeing by putting this other GRS that we're seeing a significant amount of reduction. So this can become a way for us to actually be able to start to confirm that some of these proteins that are from uncultured organisms are actually, working and doing what we think they're doing in GDGT biosynthesis. So that's where we are with this work. And I hope I've convinced you that answering a lot of the questions involved in archaeolipid biosynthesis is important, both from needing to understand just the overall structure of archaeolipids, the biochemistry and what's involved there, and how that could potentially give us insights into eukaryogenesis. Important for constraining paleotemperature proxies, as we showed in the first story that I presented, but also really important in just revealing new biochemistry and new physiology. These archaea have not been studied as well as bacteria, and we have this framework of thinking about lipids in terms of not just bacteria, but eukaryotes. And archaea just have this potential to really reveal a lot of different biochemistry and physiology. And I think it's definitely worth, even as hard as these organisms can be to work with, and as difficult as these proteins can be, we've really been able to uncover a lot of interesting biochemistry. And so with that, I'd like to thanks my group, in particular Andy and Ray, that spearheaded a lot of this work, and our collaborators in Colorado. They've been wonderful. They've been so helpful in helping us establish this new organism in our lab and the new expression system, and Brett and Valerie for sharing sequences for us and hopefully continuing that collaboration. And with that, I would be happy to take any questions.
Awesome. Thank you so much, Dr. Welander. That's a fascinating couple of stories. And I will say, as someone who works with E. coli and is used to a doubling time of 20 minutes, I can't imagine a doubling time of hours. We do have a couple of questions that are in the chat. And let me just run through them real quick, because there was some really lively discussion, and I'll also check on.
Oh wow, there's a lot in the chat.
Yeah, so and I'll also check on YouTube just to make sure that there are, if there's any there. Let's see. So this might be a little bit of a broader question. Can we see extremophiles from a perspective that they are a storehouse for important proteins to enable complex life synthesis when necessary?
That what is a storehouse?
Extremophiles, yeah.
Yeah, I mean, I don't know. I mean, extremophiles are modern organisms too, right? So, anything that, is going to lead to like more like multicellularity or evolution, or if we're thinking historically of what happened in the past, I don't think those organisms exist today. I think though, within their genomes, within maybe even their biochemistry, we can kind of get some glimpses into that. And so potentially they could provide that information, but we'd have to kind of see and understand these organisms a little bit better. And extremophiles are very broad. There's also bacteria that are extremophiles. And so, they're a very interesting group to definitely study.
Thank you. All righty. The next question we have, which I think you may have touched on a little bit, but how did you initially select the SAM enzyme as candidates for the cyclization of the GDGTs? Yeah,
that was kind of a leap of faith. Like, it was actually Roger that suggested this. Like, he was like, we all had talked about it. There had to be some kind of radical mechanism. But I think also he was the one that was like, what about these radical SAMs? Look at these radical SAMs, and started to read about the radical SAMs. There's a lot of other proteins. I mean, one of them, the main, oxygen can function to do these kinds of reactions, right? And so, but there's a lot of anaerobes that make these proteins. There's also these glycerol-radical enzymes, and those aren't really present in Archaea. So it seemed like the radical enzymes were the most likely scenario. And so, yeah, that was it was just, when you looked in the literature, there was nothing about what potentially could be making these rings. No biochemist was even attempting, because they looked at it and they said, this is just crazy. What are these microbes doing? And so we just kind of decided to focus on those. And it became kind of, because Sulfolobus had so few, it was a manageable number. And so, some of them had already been assigned function. Some of them, when we did the compared genomics, you got 3 and it was like, okay, well, deleting 3 is not that hard. So it became something that was manageable to start with. And we really got lucky. Sometimes you got to just trust your gut.
Yeah. And honestly, I feel like that's probably the most satisfying part about science, is just, okay, we're just going to take a shot in the dark and see if it works. And sometimes it works out well.
Yeah.
So there is another question that's that's pretty interesting. It has been answered in the chat, but I'll ask it anyways because it's pretty interesting. So what particulates of methanogenic organisms can help us to verify or find marker patterns of extraterrestrial life? So I know we're obviously talking about lipids here as biosignatures.
Yeah. Yeah, I think the lipids that we look at, and particularly the lipids that methanogens are making, I mean, those are very complex lipids, and I would be surprised if you can find those on, preserved somewhere outside of our planet, right? Those would be really super complex. But so the membrane lipids, of course, are a signature of Archaea, the macrocyclic archaeals, the ones that we see TES making that are kind of like those diethers, those are actually like first discovered in methanogens. And actually there's a few papers from like the 90s saying that maybe these are methanogen markers. But we know TES can make it. But now we know anything that has a TS has the potential to make that, especially if you screw up the regulation like we did with Thermococcus. So that was for a long time. But I mean, in terms of the methanogens, I mean, methane signatures, isotopes of methane, of carbon, those are always signatures that I think are going to be the most indicative of a methanogen specifically.
Awesome. And actually, this is my own question. This might be putting you on the spot, so sorry if it is. In terms of how we would detect these lipids remotely, do you have any, just out of curiosity, do you have any how do I put this? If you had a very nice grant and you wanted to detect these remotely, how would you do that?
Oh, remotely. Yeah. I mean, well, they've tried to detect, they have a mass spec on one of the rovers in Mars. So that's one way. But if you're looking beyond that, I mean, I really, the only way I know to detect these is to extract them. So return missions are going to be the way to get those. And if you're going, there's no way we're getting returns from like exoplanets. How do we actually get these out there? I mean, that's really, really hard to do. And I am someone that works on cultured organisms, right? So we do lipid extractions and from very pure cultures that we know what they're making most of the time. But when I worked in Roger Summon's lab, they had samples from rocks that were millions and billions of years old and so precious. And I just remember being able to just get as much lipid as I wanted and grow my thing up. And so I've never been short in supply, and I've been very, very lucky. I have had students who have tried to do extractions from environmental samples, and this is so, just even modern sediments are so messy for me that I'm so used to looking at just pure culture. It's really complicated. I think the hardest thing that we're going to have is that it's hard to detect these remotely. And if we can get samples, I feel like we're going to be even more than people that study like rocks. It's like there's just such a limited signal there.
Yeah, that makes sense. And again, I agree with just simply working with cultured stuff, just how quote unquote simple it can be. I'm sure. Yeah. Like I said, I'm sure with what you work with seems a lot more difficult than just working with E. coli. We do have a question from YouTube. So this one says, did you, or excuse me, have you looked at Archaeota, or do you have plans to do so?
Are those cultured? Or are we thinking of the genes? I don't know if those are cultured yet. Maybe someone in the chat can tell me. But yes, those are like there's a lot of different organisms that have GRSs that are uncultured. There was the Mars Archaeota was one that had very similar structure to the Cephalobus in terms of the genomes. So we were interested in expressing some of those as well. But yeah, the Archaeota on the list, they have some interesting GRSs. That tree that Andy generated, they all have very interesting GRSs. And we're just trying to find the limit of what is a GRS. If everything on that tree is a GRS, there's some interesting phylogenetic relationships there. And I think there's a lot. I think Andy has done the comparatives of like the 16S versus the GRS and shown that they're not congruent, that there seems to be a lot of lateral gene transfer as well.
Awesome. Alrighty, so another question. Excuse me. Are pH and temperature good predictors of GRS copy number in Archaea?
Yeah, actually, there was just a paper from Welander group that came out that looked at GRS copy number in hot spring metagenomes, and they found that it was more pH dependent than temperature. So it seems to be like it might be a pH indicator, and that might be why we are not able to get some of them to work in Cephalobus, which grows at a low pH. And we're looking at some of these from organisms that are neutrophilic that grow. And so we think that maybe something with the protein expression. And so, yeah, I don't know why those hot spring organisms have so many. And it's really interesting because they have the same lipid profile as like the marine Tom Archaeota. And so there's just kind of this interesting phenomenon within the hot spring genomes that there's a lot of GRSs.
That is pretty cool. All right, next question is, where is it? How do fresh lipid biomarkers, or fresh as in, geologic time, compare with the ancient samples?
Yeah.
Are there changes in the structure that cloud or interfere with our interpretation?
Yeah, so that's a whole area of biomarker research that I didn't touch on. That's taphonomy, right? Looking at how you can take a modern lipid, put it through the process of like high temperature and pressure and see what comes out at the end. And usually what comes out at the end is just this like paralysis mess of molecules. And so what the most basic things that you lose is you lose things like hydroxylations, amino groups, and things. Unsaturations can get lost too, but unsaturations can also, when you're going through this process of diagenesis, can then break rings and you can get these different structures. So something that say has like 5 rings can in the rock record look like it has 3 because it's broken in the middle or something like that. So there's a lot of chemical reactions that can happen. And so that's why part of this field of biomarkers, we need people who study how these break down and what are the molecules that you're doing. And Jochen Brox in Australia has been doing a lot of work on being able to analyze some of these breakdown products in the rock records, because something that we found, so another aspect of my work is we look at like sterols and these molecules, and predictions from genomic analyses tells us they should be as far back in the rock record as about 2.7 billion years, but we only see them at we see them stopping at 1.6 billion. So there's this gap of why don't we see them in there? And that's because they're probably being degraded. And what Jochen has showed is that some remnants of what we do see in this later rock record could be potentially stirring biomarkers. And that's through kind of like putting them through this, these experiments and then actually understanding what's coming out at the end. So preservation is really, really hard.
Alrighty, and then let's see, another question. Assuming eukaryotes are symbionts, why are their lipids not like Archaea?
That's the big question. I think that's one of the most difficult questions that we have, right? So eukaryotes, we have very boring lipids. Like we don't do these crazy things that the Archaea do, but there are some bacteria that fuse their membranes. There are some bacteria that make ether lipids. We make some ether lipids as storage molecules. So there are some kind of connections there that you could potentially see remnants of, but yeah, you would think that the eukaryotes would have more of a lipid of Asgard-Archaea. Now, it's possible there's an Asgard-Archaea that we haven't cultured that has a mix of these lipids, or maybe it has fatty acids and we haven't seen it. We don't see any fatty acids in Archaea. There's some reports in the literature and we don't think those are correct. And so it's just, it's a very strict divide. And I think that's years of evolution. And so it could be that some ancestor had a mix and then it was lost. That's another reason to study the lipid physiology. If you were assuming there is this transition, right? So you go, the Archaeal simia, it has these archaeolipids, and then for some reason, as eukaryotes are evolving, it gets a whole new set of lipids to make its membranes. What was that pressure? What was it about the archaeolipids that wasn't compensating for whatever it was that the eukaryotes needed? And if we understand how Archaea use their membrane lipids, when they're expressed, how they're regulated, and how they interact with their environment, could that provide some insights? I don't know. I mean, I think we're going to find some cool biology anyway if we look at that. But I think understanding archaeolipids, understanding what the potential Asgard-Archaea have for those producing those lipids, and if it's broad, I think will provide a lot of insight in that area.
Awesome. Actually, this is sort of a follow-up question. I know, at least with bacteria, there's these reports that like we've only found 1% of bacteria. I guess my question is, in terms of the difficulty that you have with cultivating Archaea, is there an estimate in terms of like how you would feel, or how much has been uncultured, and how and what would help fill in those questions? Yeah, those gaps.
Yeah, I don't know. This might be something better for like Brett to answer. I was finding these bugs all over the world. I mean, I think my understanding is that the Archaea are all over the place. They're very prevalent, but that they're maybe in lower numbers than what you see in bacteria. And that's why we haven't had this signal. I think in terms of culturing, I think I read in this book one time that the one thing that all organisms and culture share is that they are able to be cultured. That is the one thing that they share. And what is not culturable is, I think, much, at least under the conditions that we work with, is much larger than what's out there. And I think all microbiologists probably agree that we have only touched the surface of what potential in the lab of what potentially could be the diversity of microbes out there. So I think because we know that's true for bacteria, I would assume that's also true for Archaea. And I think this is coming through as more and more metagenome sequences are coming out. We're just the diversity of Archaea is insane that's out there. And so I think we're missing a lot. And I am a big proponent of bringing things into the lab and studying them, because I love to work with microbes, but I don't think we need to culture everything to understand the diversity and understand the evolutionary history of these organisms. I think a lot of the work that's coming out with these metagenome acquired genomes is really great. I think that's really been helpful. And I think, the more proteins people give me to study in the lab, the happier I'm going to be.
Yeah, I completely agree. I mean, completely agree regarding that. Alrighty. If there are no more questions in the chat, and I'll do one last check on YouTube real quick. No, we have no more questions. Well, thank you, everyone, and thank you, Dr. Welander, for joining us, and thank you for this awesome discussion regarding your work. And we're really looking forward to hearing more about looking forward to your future directions and what other questions will be answered from your work. Again, thank you so much for joining us.
Thank you, and thank you, everybody. It was great. Happy to share this work.
And feel free to, for those of you who are on our Life RCN mailing list and follow us on Twitter, keep an eye out soon regarding an announcement for our August seminar. Alrighty, thank you, everyone, and enjoy the rest of your day.
Bye. Thank you, Jaime.